Today, we will learn about the pollen-pistil interaction. In pollen-pistil interaction, pollen grains are deposited on the surface of stigma. This process starts with the pollination and ends with the fertilization process. In this process, pollination is carried out with the help of pollinating agents. Pollinating agents may be in the form of abiotic factors or biotic factors. Now here we see what is meant by pollen-pistil interaction. Pollen-pistil interaction is a sequence of all events that occur from the time of pollen grain deposition over the stigma till the time of entry of pollen tube inside the ovule. Pollination does not confirm the transfer of right type of pollen grain. Sometimes wrong type of pollen grains also land on stigma. The pistil has the ability to recognize and accept the right or compatible pollen grains of the same species, and wrong type of pollen grain is discarded by pistil. Compatibility and incompatibility of the pollen pistil is determined by a special protein. This special protein is present in the surface of stigma. This process involves the pollen recognition followed by promotion or inhibition of pollen. The stigmatic surface of flower refuse the wrong type or incompatible pollen grains, but the compatible or right type of pollen grains absorb water and nutrients from the surface of the stigma. Then these pollen grains are germinate and produces a long tube called as a pollen tube. Now this pollen tube is finally pushed through the ovule and reaches this embryo sac. Inside the embryo sac, there are 7 cells and 8 nuclei present. When this pollen tube reaches the embryo sac, the tip of pollen tube enters in one of the synergid cells and then ruptures to the synergid cells to release the content. Due to the pollen-pistil interaction, intense competition develops even in the compatible or right type of pollen grains. It also plays a very important role in the sexual reproduction and seed formation.